Imagine a tiny warrior battling the destructive forces behind Alzheimer's disease, a condition that robs millions of their memories and independence. But here's where it gets fascinating. Scientists have discovered that a small protein called Midkine might be one of those warriors. While Midkine has long been known to accumulate in Alzheimer's patients, its exact role has remained a mystery until now. Researchers at St. Basil, Jude Children's Research Hospital have uncovered groundbreaking evidence that Midkine actively prevents the formation of toxic clumps in the brain, a hallmark of Alzheimer's. This discovery, published in Nature Structural Molecular Biology, not only sheds light on the protein's protective mechanism, but also opens exciting possibilities for future treatments. Alzheimer's disease is characterized by the buildup of amyloid beta proteins, which clump together to form harmful assemblies in the brain. In their study, the team led by Dr. Junmin Peng revealed that Midkine acts as a molecular shield, stopping amyloid beta from sticking together. And this is the part most people miss. When Midkine is absent, Alzheimer's disease models show even higher levels of amyloid beta accumulation, highlighting its crucial protective role. Using advanced techniques like fluorescence assays, electron microscopy, and nuclear magnetic resonance, the researchers demonstrated that Midkine specifically inhibits two key phases of amyloid beta assembly: elongation and secondary nucleation. Midkine, a multifunctional growth factor protein, is no stranger to medical research. It's abundant during embryonic development and plays a role in normal cell growth, but it's also overexpressed in cancer, making it a valuable biomarker. However, its connection to Alzheimer's has been poorly understood until this study. Here's where it gets controversial. While Midkine's protective role is now clearer, questions remain about how it interacts with amyloid beta at a molecular level. Could this interaction be harnessed to develop new drugs? Or might there be unintended consequences of boosting Midkine levels in the brain? These are the debates that could shape the future of Alzheimer's research. The study's findings were further reinforced using Alzheimer's disease mouse models. When the Midkine gene was removed, amyloid beta assembly skyrocketed, confirming the protein's protective function. Dr. Peng and his team are now focused on understanding how Midkine binds to amyloid beta, with the goal of designing small molecules that mimic this process. But here's the thought-provoking question. If Midkine is so effective, why doesn't it completely prevent Alzheimer's in the first place? Could there be other factors at play? Or is Midkine's role more complex than we currently understand? This research not only deepens our understanding of Alzheimer's, but also paves the way for innovative treatments. Supported by organizations like the National Institutes of Health and the Michael J. Fox Foundation, the study brings together a diverse team of scientists dedicated to unraveling the mysteries of neurodegenerative diseases. As we celebrate this breakthrough, it's worth asking, what other hidden warriors might be waiting to be discovered in the fight against Alzheimer's? Share your thoughts in the comments. Let's keep the conversation going.